Okay. So as we move on in our story on inflammation, we have a whole lecture on inflammation, which is pretty phenomenal, but it's important because that's a big part of what we're going to be covering this semester. There are different types of inflammation, like we have different types of necrosis. And so if we look at the different types of inflammation shown on this slide, we can categorize them based upon their features. So we look at serous inflammation. There's a lot of effluent or effusions. That means there's fluid in the cavity, typically in a serous cavity. You're going to get a watery, a protein-poor fluid. So it's not exudate, it's basically just transudate. A great example of a serious inflammation would be a blister. You know how the blisters have kind of that bag of fluid. Okay, but it's not pus. It's like if you lice it, some of you are that way. You can't not touch it, right? How many of you are licers? Yeah, there you go. How many of you just leave it alone, right? People that never had a splinter, right? Like, don't touch it, don't touch it. It's full of fluid or something. Somebody just can't help it. Like, oh yeah, right? And it's like a proteinaceous, watery, clear fluid that comes out. Fibrinous. Fibrinous happens with a more extensive injury. You get larger capillary permeability. You get a coagulum. You get a lot of eosinophils migrating into the area. And it's typically seen with inflammation associated with body cavities. Fibrinous. Superlative. Superlative is where you get an exudate. That's like a white pus that comes from neutrophils. The white pus is usually necrotic cells and edemic fluid, but you got a lot of cellular debris there, and that's why it's kind of like a pussy fluid. Different than a blister. In other cases, it may be caused by an infection. So pyogenic if it's a microbe. And then abscesses are, like an abscess in a tissue is a local accumulation of pus that's been kind of walled off by the body. Okay, that abscess. And so if you come in and surgically remove it, it will be pretty nasty inside, foul smelling. Okay. And then the last type of inflammation is an ulcer. And we'll go over the pictures here in a second. But the ulcer is usually erosion of an organ or a tissue due to the shedding of necrotic cells. So what do we see on the pictures? In the lower left is a duodenal ulcer. And that should be fairly obvious after I tell you that. Because you can see here is the epithelium that normally should come right across like this. And this is the ulcer that's formed into the underlying or deep tissue, duodenal ulcer. On the lower right is a purulent inflammation of the lung. And what you're seeing is an abscess that contains neutrophils and other cellular debris. That's right here. So all of these purple dots are these transmigrated neutrophils. There are the first responders, cell type. The first cell type that shows up are the neutrophils. And they got there first. And all of these red network structures are capillaries. That's where they came from. So normally, this right here would all be the color of this white space. This would all be open. And it's filled with all these inflammatory nuclei. So that is a purulent inflammation of the lung. Now the upper right, this is a fibrinous inflammation. This is pericarditis over the heart. Pericarditis. So you see the underlying heart, and this is the pericardium that's been pulled back. And you can see excessive amounts of fibrin deposited on the pericardial tissue. That fibrinous deposition is all that white, glistening stuff. All this white, glistening tissue here shouldn't be there. Okay, so different types of inflammation. Now, when we get inflammation, we said in the very beginning that it's a good thing. And we have a couple of different types of outcomes. There's basically three. So we can either stimulate an acute inflammatory response due to an injury. You see the different types of injuries. We could have trauma. We could have a toxin that shows up in the tissue. We could have infections for bacteria. Or we could have an infarction, which is a loss of blood flow. You can have an infarct to brain tissue, we call it a stroke. We have an infarct to heart tissue, we call it a myocardial infarction. We have kidney infarcts, splenic infarcts. There's infarcts of all different types of tissues. Now, that would cause an acute inflammatory response like we just studied in detail. From here, we could actually resolve the wound like we saw in the nice pretty graphs, and everything returns to normal function, and that's called resolution. Or, we actually may see it progresses and it never resolves because the injury sticks around and it's constantly there. And this would be in the category of chronic inflammation. Okay? So upper right, which is our favorite, and then the lower left, which is our least favorite. And in chronic inflammation, you can see some of the causes of chronic inflammation because these are things that don't subside. They don't ever really go away. Viral infections of that particular tissue bed. Or some sort of chronic infection that won't resolve. Persistent injury that takes place, like what we saw the first day of class with Barrett's esophagus. That's a persistent injury that could be a chronic insult to the tissue and stimulate chronic inflammation. Or various autoimmune disorders, like lupus or MS. Okay? What's going on over here, though? Okay, well, the third one would be a healing response instead of wound resolution. This healing response is you don't have normal function returning, but you can see the blood vessels don't stay dilated. They actually subside. They come back to a normal phenotype. But the surrounding tissue is scarred. You don't have a return of normal function in that tissue. Now you replace it with fibrotic tissue, which is scar tissue. And that's what you see on your skin. You're going to study that next week with tissue healing. But a fibrotic wound is scarring after inflammation has subsided and your tissue has been reconstructed, but it doesn't get reconstructed in the same capacity and you don't have normal function. So the example would be like if you had a big injury on your head and that little area, the hair never grew back on that particular region. Well, you know that the skin has hair cells in it, and so normal function of skin would mean all of the sebaceous glands, all of the American glands, all of the hair follicles would regenerate and grow back. You can see that on your arm. Maybe there's a big scar on your arm where even the hairs on your arm didn't grow back, and the tissue, you can feel it's stiffer. It's more rigid than the surrounding tissue, which is more supple or normal feeling. Okay? So this is a fibrosis response that we see, and if this is in heart tissue, we call that the scarring in the myocardium, and that particular portion of the heart no longer moves. You get no longer to get cardiac motion in that region because it's a stiff fibrotic scar.
Like, we've always been told that we scar because we heal too quickly. Is that still correct, or why do we scar? Like, why does that not heal correctly?
The reason that we scar is because the cells that originally were available to regenerate that tissue no longer function in that capacity.
Okay.
So for example, in the embryo, we get scarless wound healing. And we know that because we've done surgery as a field, not me, but the field has done surgery on spina bifida babies. They have really messed up spines, and they come in surgically in utero, make the correction, and they hardly have an incision when they're born. It's unbelievably nice. And so we know that wound healing that takes place in the embryo is considered scarless wound healing. And as soon as we're born, we lose that capability.
Do we know why? Just because all the...
We have a lot of theories. One of the big ones, this is a little side tangent, it's a little bit more next week, that's okay. One of the big theories is that the embryo is a low oxygen environment, it's hypoxic. And there are certain genes that are on in a hypoxic environment that tell the cells to do certain things, that the moment you take your first breath, you turn those genes off because the organism is no longer hypoxic. And one of them you can research is called HIF1 alpha, hypoxia inducible factor alpha. So if you want to do some research on HIF1 alpha, and wound healing, it's pretty fun. Okay? So this is fibrosis. Now what about this one? This is kind of an intermediate. That's why it doesn't say 4. It only says 3. This one is where you get an intermediate step before it moves to fibrosis, where you have a lot of pus formation as you're cleaning up the cellular debris. But it eventually will move on to this fibrotic scarring where you have loss of function. It will not, you see the arrow doesn't go up this way. It will not go this way. Okay? So there's really only 3 main outcomes. Complete resolution, you get normal repair of function. Some of you may have cut yourself when you were very young. You fell off your bike, you had this massive spill, it was gnarly, and you can barely find the scar. Because when we're younger, our healing is more like embryonic healing than when we're older. And then some of you cut yourself last week on a sheet of paper, and you think you might have to amputate that finger because it looks so, right? So as we get older, it only gets worse, guys. As you get older, your capability of normal function returning to that repair is compromised. All right, so now we're transferring into chemical mediators of inflammation. And this is the point in the lecture where I start glazing over because there's 3 slides of like heavy mechanistic pathways. Okay, so I'm going to go over the 3 slides of heavy mechanistic pathways, and then I have a summary slide that kind of wraps it all up. Okay, for our non-splinter people that don't like to intuit too much. So where do the chemicals come from? You keep talking about all these chemicals, right? Like histamine. And you said they came from mast cells, and that's like all you said. Well, here you go. That's you, and you want to know more. Here's the complete story, okay? So we basically, at the site of injury, we have 2 main sources where these chemicals can come from. They can come from the injured tissue itself, right? Or they can come from leukocytes. The leukocytes could have them already ready to go off the shelf, like in inventory. Those would be preformed chemicals that are actually sitting in vesicles inside the cell. And that would be things like histamine and serotonin. All they have to do is fuse the membrane and dump the contents. Or the cells could actually synthesize them from scratch, which is called de novo chemicals, and they manufacture them. And here's the list of things that they manufacture, and here's the source of the cells that can do that. So let's just take a couple for example. If we look at histamine and serotonin, these guys, histamine comes from mast cells, basophils, and platelets, and serotonin comes from platelets. If we look at some of our synthesized materials, like prostaglandins and leukotrienes, we're going to talk about those next. Platelet activating factor, or reactive oxygen species, these all come from all leukocytes, including macrophages. We went over that. We also went over nitric oxide, which comes from macrophages and endothelial cells. And then we have general categories like cytokines and neuropeptides that can come from macrophages, lymphocytes, endothelial cells, mast cells. Even nerve fibers could be releasing these neuropeptides that have capability of causing vascular modifications. Now, we also have the ability to create stuff from the liver. And we put these sources into the bloodstream to do certain key things as a part of inflammation. So there was a question at the break about how do we get margination and why does vasodilation cause the cell to move to the edge? Well, that's not the whole story. The second piece of that story comes from the kinin system and coagulation. And so I'm going to tell you that here in a moment. Okay? So if we look at cell-derived mediators on this slide, and this is very busy, but let's start at the top and walk through it systematically. We start at the top. These cell-derived mediators come from a family known as eicosanoids. Eicosanoids are a family of molecules that are all created from arachidonic acid. And arachidonic acid is actually manufactured from the phospholipid bilayer. So here you see right here in the middle top arachidonic acid. And where did it come from? It came from the phospholipid membrane. And you used a phospholipase, ACE, an enzyme, to break down the lipid membrane to make the precursor arachidonic acid to manufacture all this stuff down here. Okay? So arachidonic acid is your starting material. Where'd you get your starting material? It wasn't from Safeway. Okay? It was from the membrane of the cell, which is convenient because it's sitting around. Well, arachidonic acid can go either cyclooxygenase direction or a lipooxygenase direction. Both of these are enzymes. They break it down further. And cyclooxygenase basically will manufacture the family of prostaglandins, and that goes all the way to the bottom to give you this action down here of vasodilation and increasing vascular permeability. It makes the family of prostacyclins, which causes vasodilation, and it inhibits platelet aggregation. So you don't get a bunch of clotting showing up and stopping blood flow altogether. It stimulates, via this pathway, cyclooxygenase, thromboxane family, which causes some vasoconstriction and helps to promote platelet aggregation. So why do you get both vasodilation, vasoconstriction, inhibiting and promoting, is this is the idea of a controlled coagulation, or a gentle coagulation. It's hemostasis, which the word means blood stagnant, but it's temporary hemostasis that's under a controlled fashion because you're releasing sort of these boundary markers that say, don't go too far. We don't want to shut off blood flow, for crying out loud. We just want to get these cells to move towards the edge, get sticky, and start rolling, and then migrating out. Does that make sense? So your cyclooxygenase tree, this side, prostaglandins, prostacyclines, and thromboxanes. So you need to know the pink boxes, and you need to know what side it came from, and ultimately where you got it from. Now, the next side, your right, is using a different enzyme known as lipooxygenase, which takes arachidonic acid and makes it into this family of leukotrienes. And the family of leukotrienes basically give rise to vasoconstriction and an increase in vascular permeability. Again, another checkpoint for the vasodilation, but it still allows for permeability to go up. And a byproduct of this also helps to stimulate chemotaxis. So this is where a lot of these cytokines that we keep saying come from. We say them very generally, and then later on we smack you over the face with all of the heavy details. Okay? Now, the last mechanistic slide, I lied, there's two. So there's four total. So the third of the four mechanistic slides, so they're plasma mediators. Okay? These plasma mediators are made by the liver and put out into the circulation. And the family of these plasma mediators are called complement. And what I want you to know about complement is the complement system is this biochemical cascade that helps or complements, if you will, and that's why it's called that, the ability of antibodies to get rid or clear pathogens. It's part of the immune system. It's called the innate immune system because it doesn't change. The complement proteins are all the same. It consists of a number of small proteins that are made in the liver and they're released into the bloodstream. And when they're stimulated, they actually cause proteases in the system to cleave specific proteins so that they release other cytokines and allow new cascades to take place. So let's take a look here. So this is complement C3b. This is complement C3a. So you can see there's three main pathways that I want you to understand. There's complement factors that allow for a increasing of capillary permeability and vasodilation, leukocyte activation, and phagocytosis. These three pathways are on the left. They all converge into the middle. They all end up allowing for all three of these outcomes to take place. So on the far right is really the take-home message, which is C3a and C5a stimulate an inflammatory pathway, where you can see they bind to the leukocyte, and in so doing, they make it more effective in phagocytosing a microbe. Okay? Here, the C3b pathway does a similar process, independent of inflammation though, where it facilitates phagocytosis by allowing the microbe to be seen by the phagocyte. And you see this 3CB complement is what's allowing the binding to happen. And then the last one is the formation of what we call the membrane attack complex, where you get the organization of complement proteins into the microbe to form a channel, and that channel basically puts a hole right in the membrane and allows for the membrane to be lysed. Okay, so they're complementary. The last mechanistic slide, and I'm correct on this one now, and the last mechanistic slide is looking at some additional plasma mediators that are part of two main pathways, the kinin pathway and the coagulation pathway. So if we look at the kinin pathway on the left side of the screen, we start out with Hagman factor, also known as factor 12. You guys remember the coagulation cascade from 202 and blood clotting?
Remember of it.
Yes. Okay. So factor 12, Hagman factor, we call it a cascade because factor 12, let's do the clotting cascade first. Factor 12, otherwise known as Hagman factor, is converted to factor 12A. And it's converted and activated by platelets or by exposure to collagen, which is found in the base of the membrane. So if you nip the skin, you expose collagen to the blood. And then Hagman factor goes from 12 to 12A. Make sense? So 12A is the stimulus of taking 11 to 11A. So on and so forth until you get down to thrombin, which is 2A, takes fibrinogen to fibrin. And this actually is what makes up the clot. So it's a cascade, that's why it's called a clotting cascade. It starts with factor 12. So why do you care about the coagulation cascade as it relates to inflammation? Why is it important? What does hemostasis allow for? On the edge of the blood vessel during inflammation. What's that? It's a controlled coagulation, right? Say that 5 times fast, right? That allows for the margination of those cells to go to the edge. Now, if we back up to factor 12, it's also the beginning point for the creation of 12A, which doesn't just stimulate the coagulation cascade, it also stimulates the kinin cascade. The kinin cascade, again, more complicated. I don't care as much about these intermediates. What I care about on the kinin cascade, as well as the coagulation cascade or clotting cascade, is you know what comes in and you know what comes out. What comes out of the kinin cascade is plasmin, which actually activates fibrin, plasmin, which actually splits up fibrin, so it shuts it down. Does that make sense? So this is the off mechanism of the coagulation. That's important because you don't want to just We said it's controlled because you stimulate both pathways. Here, plasmin also activates C3 to C3A. Now you get the complement activation that we looked at on this slide. Okay? So what does this look like if we summarize it? I made a summary table for you because I know that there's a lot of moving parts in the last 4 slides. And these are what I would consider to be the most salient points, not necessarily the only ones I'd ask questions about, but they are the top level points over the last 4 slides. With vasoactive amines, histamine, increasing vascular permeability, and allows white blood cells to exit tissues. Serotonin regulates homeostasis and blood clotting. Eicosanoids, you got your 4 families of eicosanoids, all that come from arachidonic acid, and they have vasoactive effects, whether it's vasodilation or vasoconstriction and chemotaxis. Complement, which supplements or complements phagocytic and inflammatory pathways. And last but not least is our kinin coagulation pathways, which allow for and also control hemostasis, which we just said is important for encouraging those white blood cells to move to the edge. Right? That helpful? Okay. So in the remaining time, we need to apply this to chronic inflammation. You're like, how's he going to do that in 7 minutes? Well, here it is. It's pretty much exactly what we just talked about for the last hour and 53 minutes. Except it never stops. Okay? So chronic inflammation uses all the same pathways and steps as acute on the left. But there's a big difference. So for example, you got inflammation coming up and coming down. Right? We said 10 days, right? I told you histologically about 14 is our cutoff where we're looking very carefully. We can't see it anymore. Then we got phase 2. Phase 2 is where we get cell proliferation and matrix deposition, and it comes down. And then matrix remodeling, phase 3, goes out to, even almost a year after the injury. And you know that. I mean, you've seen a really aggressive scar, even 6-9 months later, still a little pinky maybe, not quite there. About a year, 2 years later, it's done, right? It's not going to change much. It's not going to fade anymore, right? So a lot of the anti-scarring ointments target this phase of wound healing. But if it's older than a year, there's probably not much you can do for it, okay? Now, if you look at chronic, we get our neutrophils coming up and going down, like we'd expect over here. But look, we get macrophages staying up and elevated. We get blood vessels sticking up and elevated. We get these things called foreign body giant cells that I've never told you about. We're going to talk about those in a second. Fibroblasts stay elevated. Fibrosis stays elevated. Stuff just keeps going on in a chronic nature. Now, histologically, if we look at some of the differences that are shown here, the top is a chronic inflammatory reaction, okay? And this is in the lung. The collection of chronic inflammatory cells is denoted by the asterisk. And you can see that right here. That's chronic inflammatory cluster, all the purple dots. The arrows themselves are fibrosis. This is all fibrotic scarring right here. Now these arrowheads, the arrowheads are actually normal alveoli spaces are replaced by cuboidal epithelium. Okay, right here. You get metaplasia taking place. Now, in acute inflammation down low, you can kind of see that what's happening here is the outline of the alveoli, and you've got inflammation here, but in contrast, the collection here, these are all neutrophils. Neutrophils, here's a collection of neutrophils. Still purple dots, but they don't get together as close and cluster as tightly as what you see right here. Can you appreciate that the tightness of the clustering of the macrophages up here is tighter than what you see down here with the neutrophils? Make sense? So with chronic inflammation, you're going to get a lot of infiltration of neutrophils. They'll go away. Macrophages will come in and they'll stay there for a long time. In fact, what happens is they stay there so long that they start to fuse. And as they fuse together, they form foreign body giant cells, which are multinucleated. Multinucleated foreign body giant cells. And we'll talk about that here in a couple slides. So, at the level of chronic, we still have neutrophils as our first responders. We still have monocytes that become activated tissue macrophages. But now we start recruiting other cell types, which was mentioned earlier. These other cell types are B and T lymphocytes. And we're going to talk about that in 2 weeks as we look at what these T lymphocytes can do, these B and T lymphocytes can do in a chronic inflammatory situation. We also stimulate more mast cells and more eosinophilic infiltration. So the last histology of the lecture is showing foreign body giant cells. So here you've got classic chronic inflammation, because you can see out here in the periphery this walling off of this granuloma. The granuloma is in here. This is not going to go away. It's too big for these collection of macrophages in a chronic inflammatory response to come in here and phagocytose it up. So what do? They start joining forces and fusing into these giant cells. These are called foreign body giant cells. They're multinucleated and very, very large. You can see them with the blue arrows. Okay, there's one there, there. There's one here, and one here. There's also one right there. So classic chronic inflammation is the presence of macrophages, foreign body giant cells, and then this fibrous capsule as you wall off that local infection. Okay? The fibrous encapsulation, the foreign body giant cells, the walling off of that chronic inflammatory event are characteristic signs of chronic inflammation versus acute inflammation. Yeah?
Why is that walling off? Is it like an accident? Because that can't help it heal. So like.
It's not an accident. It's the well, if we can't get rid of it, we're just going to wall it off so it doesn't do anything else.
Like spread, okay.
Okay? So the last slide for today, and let me explain what's going to happen with the rest of the day. The last slide. This right here, this is a granuloma. A granuloma. So you would find a granuloma in various different types of tissues, but you'd find it, for example, like in tuberculosis in the lungs. Okay. The last histology slide that I want to show is how this applies to even some of the research that I do. So this is a biomaterial known as polypropylene. And it's a very inflammatory material. This was implanted, okay, into tissue. Here's the explanted tissue here. You can see how red it is. And this is the material, this white space. And you see all of the red arrowheads? Those are all foreign body giant cells. So when we take out the tissue with regular old surgical polypropylene, we know that it's a chronic inflammation. And what this project is how can we take polypropylene and how can we actually make it less inflammatory so that it heals better, so it's not chronic inflammation. So we coated it. We used this proprietary coating, and this is the same material coated, and you can see this tissue is far less angry. There's truth, is hard to ignore. This is a foreign body giant cell, but the relative number of foreign body giant cells here is far fewer than what you saw over here. So this is a way that coatings are being used to mitigate chronic inflammation and to promote healing in a better way. Okay? So that is the last slide of the deck for the exam. There's additional slides back here that talk more about this biocompatibility with biomaterials for those students that are interested, but I will not ask test questions after this last slide. So this is the last slide that would be pertinent for the class, and everything beyond here that's available to you is more information about that topic. It's a little bit of a side tangent, so that's why I don't go into it. Okay? Fair enough? Have a great next week. I'll see you in 2